Hello, everyone. Today we're diving into a crash course on the working cell.
It's so easy, when you first learn about biology, to picture the cell as just this random bag of parts.
But that mental image is, well, it's fundamentally wrong.
It really is. The living cell isn't some museum piece. It's a bustling, nonstop factory. It's a masterclass in highly sophisticated systems integration.
And these are, they're multiple self-contained operating systems that all have to function together perfectly all the time.
And to really get a handle on the cell's genius, we find it helps to break its operation down into three core interdependent systems.
Okay.
So system one is what we call the dynamic interface and border control, basically how the cell manages its boundary and controls all the traffic coming in and out.
Then system two, that would be the power grid, the universal currency system. This is what keeps the lights on.
Exactly, the energy transformation. And finally, system three is the specialized workforce and assembly line. This is all about enzymes and the very precise management of chemical reactions.
And you really can't understand the cell until you see how these three systems are constantly, relying on each other.
So let's start at the edge, at the perimeter, with system one.
The dynamic interface. We should probably start with the blueprint for this boundary, which is called the fluid mosaic model of the plasma membrane.
The key insight here is right there in the name. It's not a rigid wall, it's a living, moving fluid.
Right.
It's primarily what we call a phospholipid bilayer, so two layers of these fatty molecules. But the important part is that those molecules are constantly shifting, creating this dynamic surface.
And it's that specific structure that gives the membrane its most crucial characteristic: selective permeability.
Yes.
It's the ultimate border guard. It determines exactly what gets to cross: ions, hormones, water, everything.
And that fluidity part of the model is just critical for survival. That movement is maintained largely because of these little kinks in the unsaturated fatty acid tails.
Ah.
Those kinks prevent the phospholipids from packing together too tightly. You can think of it like a crowded dance floor where, everyone is just a little bit slippery. You can't lock into a rigid formation.
And in our cells, in animal cells, there's also cholesterol, right, tucked right in there.
Yes, exactly. And cholesterol is, well, it's sort of the ultimate consistency regulator. It prevents the membrane from becoming too fluid at high temperatures or too stiff when it gets cold. It's a buffer.
So that's the fluid part. What about the mosaic?
Now, the mosaic piece is where the real action is. This refers to the huge, diverse array of proteins embedded within that membrane. Transport proteins, glycoproteins, receptor proteins.
And these are the true functional parts, the specialized gatekeepers and communication hubs.
Precisely. And speaking of those hubs, their roles are just astonishingly diverse. We can kind of group them into three buckets: movement, communication, and structure.
Okay, so for movement.
For movement, you have transport proteins. They act like these custom-built tunnels or ferries, allowing specific molecules to pass through.
And communication.
For communication, receptor proteins are absolutely essential. They don't let things in, but they bind to signaling molecules outside the cell like a hormone. And that binding relays a message inward, which then triggers a response inside the cell.
And you mentioned structure as well.
Right. Some of these membrane proteins actually link the cell's internal scaffolding, the cytoskeleton, to the outside world, the extracellular matrix.
Wow, so it's anchoring the cell in place.
It lets the cell sense tension, coordinate movement, and anchor itself properly within a tissue. It's incredibly sophisticated engineering.
So this complexity brings us to the actual strategies for molecular traffic. How do things actually cross this border?
Right. It all really boils down to a simple question. Are molecules moving with the natural flow, or does the cell need to fight against it?
Moving with the flow, that's passive transport.
Yes, and it requires zero energy from the cell. Molecules are simply moving down their concentration gradient.
From where there's a lot of them to where there are fewer.
Exactly. The simplest form is diffusion. This is how you get oxygen into your cells and carbon dioxide out. Because O2 and CO2 are small and nonpolar, they can just sneak right through the lipid bilayer.
But most things the cell needs, like glucose, ions, amino acids, they're either too big or too polar to do that.
For those, the cell uses something called facilitated diffusion. They still move passively down the gradient, but they need help. They require a specific channel or a carrier protein in the membrane to get across.
Wait, hang on. Why would the cell ever need to fight against that natural flow? It seems so inefficient, moving things where they don't naturally want to go.
It does seem inefficient, but it's absolutely vital for maintaining life itself. This is active transport. It requires a ton of energy, usually from ATP, because the cell is intentionally moving molecules against their concentration gradient.
Pushing them uphill.
It's exactly like pumping water uphill. A nerve cell, for example, needs a much lower concentration of sodium inside than outside to be able to fire. So it has to use these specialized pumps to actively push sodium out, constantly fighting diffusion.
So that ability to maintain an internal environment that's completely different from the external one, that's what makes complex work possible.
It is. And since water is so fundamental to all this, we should touch on tonicity.
Which is just the ability of a solution to make a cell gain or lose water.
Exactly. We tend to focus on the extremes. A solution that makes a cell shrivel is called hypertonic. It has a higher solute concentration, so water rushes out of the cell.
And a solution that makes it swell is hypotonic lower solute, so water rushes in.
Now, here's where the difference between animal and plant cells is a really key insight. If you put an animal cell, like a red blood cell, into a hypotonic solution, it has no protection. It just bursts. It lyses.
But a plant cell is different. It's got that rigid cell wall.
Right. So in that same hypotonic solution, the plant cell swells up, but the wall pushes back, preventing it from bursting. This state, called turgid, is actually the normal, healthy, firm state for a plant. It's what holds them up.
What about the other way? In a hypertonic solution?
If you put that same plant cell in a salty hypertonic solution, the water rushes out and the plasma membrane actually pulls away from the cell wall. That condition is called plasmolysis, and it's lethal.
And this isn't just some abstract theory. You can see this at work in your kitchen.
Oh, absolutely. Preserving food by salting meat works because you're creating a powerfully hypertonic environment. Any bacterial cells on the meat they plasmolyze and die. It prevents spoilage.
It's chemical warfare on a microbial scale.
That's a great way to put it. So that pretty much wraps up our border control system. Now we can transition from, managing the boundary to actually powering the whole operation.
Okay, so let's shift focus to system 2, the power grid.
Cellular operations, everything from transport to building new molecules, is all governed by the same two laws of thermodynamics that govern the entire universe.
The first law is the conservation of energy. Pretty straightforward. Energy can't be created or destroyed, just transferred.
Right. A plant takes light energy and transforms it into the chemical energy stored in a sugar molecule.
The second law, though, that's the one that's a bit of a headache. Every single energy transfer increases the disorder or entropy of the universe.
Some energy is always lost as unusable heat. It's an unavoidable tax on every transaction.
And that heat loss is a good way to measure efficiency, right?
A great way. When your car burns gasoline, only about 25% of that chemical energy actually turns into kinetic energy to move the car. The other 75% is just wasted as heat.
And the cell.
The cell is a bit better. Cellular respiration captures about 34% of the energy from glucose as useful energy for work, but the remaining 66% is lost as heat.
So next time you're sweating through a tough workout, you can remember that you're basically just a high-tech furnace burning off the entropy of the universe at 66% inefficiency.
That's one way to look at it. And the sum of all those chemical reactions inside that furnace is what we call metabolism. We can split these reactions into two types, based on whether they release energy or consume it.
Reactions that release energy are exergonic energy outward.
Yes, they're naturally downhill reactions. The reactants start with high potential energy and end with products that have low energy. Cellular respiration, breaking down glucose, is the classic example.
And the opposite would be endergonic reactions, energy inward, which need a net input of energy. They're uphill.
Right. Photosynthesis is the premier example. It builds sugar from CO2 and water, but it needs light energy to do it.
The real genius of the cell, then, is in what's called energy coupling.
Yes. You use the massive energy release from exergonic reaction like breaking down your food to immediately drive the endergonic reactions that build molecules, create structures, and do work.
And the molecule that acts as the universal currency for this, the one that pays for almost all cellular work, is ATP, adenosine triphosphate.
Its power is all in its structure. ATP is made of adenosine and a tail of 3 phosphate groups. And each of those phosphate groups carries a negative charge.
And like charges repel.
Exactly. So those charges are all crowded together, desperately repelling each other. You can think of it like a highly compressed spring just packed with potential energy.
And that energy gets released when the final phosphate bond is broken. ATP becomes ADP plus a phosphate group.
Crucially, that energy doesn't just float away as heat. It's transferred directly to other molecules in a process called phosphorylation. That transfer energizes them to do work.
Whether that's chemical work, like building a protein, or transport work, like we saw with the active transport pumps.
Or even mechanical work, like the motor proteins that make your muscle cells contract. ATP fuels all three.
The speed of the ATP cycle is just, it's mind-boggling. It's completely renewable.
Constantly. Energy from exergonic processes, like respiration, is used to synthesize ATP, which is an endergonic process. Then the breakdown of that ATP fuels everything else.
I read that a single working muscle cell regenerates about 10 million ATP molecules
Yeah.
every single second,
It's an unbelievably high-speed economy.
which means it must need absolute control.
Mm.
And that brings us to System 3, the specialized workforce enzymes.
Exactly. The fundamental problem for the cell is stability. I mean, if complex molecules like DNA and proteins are so energy-rich, why don't they just spontaneously fall apart?
They're stable because of something called the activation energy barrier. It's the energy required to get any reaction started. It's like a kinetic lock.
Right. And if the cell just used heat to get over that barrier, it would speed up all reactions indiscriminately. It would be total chaos. You'd cook the cell.
So the solution is enzymes.
The brilliant biological solution. Enzymes are reusable catalysts, almost always proteins, that bypass the heat problem. They dramatically speed up specific reactions by selectively lowering that activation energy barrier.
Allowing only the necessary reactions to happen, and to happen quickly.
And the whole world of enzyme function comes down to that core biological theme. Structure determines function, and enzyme's specificity comes entirely from its unique 3D shape.
The specific molecule an enzyme acts on is called the substrate.
Mm-hmm. And the substrate slots into a specific pocket on the enzyme called the active site.
Now, the key mechanism here is the induced fit. It's often taught as a lock and key, but that's a little too static, isn't it?
It is. Induced fit is more like a dynamic handshake. The active site actually changes shape slightly as the substrate binds, sort of embracing it more snugly,
And that physical embrace contorts the substrate's chemical bonds.
making them much easier to break, and that's how the reaction accelerates so dramatically. Once the substrate is converted to products, say sucrose into glucose and fructose, the products are released and the enzyme emerges totally unchanged, ready for the next round.
Which explains how a single enzyme molecule can process millions of substrates per minute.
But with reactions happening that fast, the cell can't just let them run wild. It needs exquisite control. And that regulation comes from molecular inhibition.
An inhibitor is basically a chemical that interferes with the enzyme.
Right, and there are a couple of main types. Competitive inhibitors look a lot like the actual substrate. They compete for the same spot, effectively blocking the active site.
And then there are non-competitive inhibitors, which I think are fascinating. They don't even go near the active site.
No, they bind to a completely different location on the enzyme, but when they do, they change the enzyme's overall shape.
Which distorts the active site. It breaks the enzyme's ability to work.
And the cell uses this strategy internally through feedback inhibition. This is a brilliant management concept where the final product of a metabolic pathway, once it builds up, loops back and inhibits an enzyme early in that same pathway.
It's the ultimate, we have enough, stop production signal. It prevents waste.
Which brings us to the final, big picture, how all three of these systems cooperate. Let's look again at active transport.
Okay, so the transport protein itself, that's embedded in the membrane. That's system 1, the interface.
Right. Then system 2, the power grid, fuels the operation. ATP delivers the energy directly to that protein.
And the protein itself acts as the workforce, system 3. It acts like an enzyme. It catalyzes the breakdown of ATP and uses that energy to change its own shape.
That phosphorylation and the shape change provide the mechanical force to pump the solute across the membrane against its gradient. It's a perfect, instantaneous cycle of membrane control, energy usage, and enzyme function, all in one.
And finally, let's just touch on the cornerstone application of understanding all this. Modern pharmacology, human health.
So many of our most successful modern medicines are just elegantly designed enzyme inhibitors. They target a specific biological worker to disrupt a disease.
Like ibuprofen.
Exactly. Ibuprofen works by inhibiting an enzyme that produces prostaglandins, which are messenger molecules that cause pain and inflammation. You block the enzyme, you block the pain signal.
Antibiotics, too. Penicillin blocks the active site of a bacterial enzyme that's vital for building its cell wall. Without the wall, the microbe just bursts.
And in viral treatments, drugs like HIV protease inhibitors target a key viral enzyme that the virus needs to mature and replicate.
That precision targeting is what makes modern medicine so effective. But it also highlights the danger of things like nerve gases or pesticides.
Yes, because they often use irreversible inhibition. They bond permanently to critical enzymes, causing a catastrophic systemic shutdown that the body simply can't recover from.
So we've covered the cell's three dynamic, interdependent systems: the selectively permeable membrane, the ATP energy currency, and the precise control offered by enzymes.
It's really just mind-boggling when you stop and think about the efficiency. We know the second law of thermodynamics is constantly pushing the entire universe towards disorder, towards chaos.
Yet, inside every living thing, trillions of cells are using these precise enzyme-driven reactions, fueled by millions of ATP molecules every single second.
They're creating and maintaining this incredible, localized, low-entropy order out of chaos.
So as you go through your day, maybe consider this profound fragility and resilience. What subtle, high-speed, enzyme-driven process is running in the background right now that prevents the complex chemical system that is you from simply collapsing into universal disorder?